health lovers. So it's your cute student nurse V, and today we are going to be talking about urine formation. So how is this urine formed? Urine is formed in three stages. Now we have the glomerular filtration, we have the tubular reabsorption, and we have the tubular secretion. Now let's dive into the glomerular filtration. Now the glomerular filtration takes place in the glomerulus, where the afferent arterioles goes into the Bowman capsule with full pressure. Now it's going in with much pressure because it has a lot to be filtered. Now there are two types of filtration that takes place there. We have the selective filtration and we have the ultrafiltration. So the selective filtration is the filtration of small solutes like water and other small solutes. Now the ultrafiltration has to do with when large molecules are not being filtered due to their molecular weight. So large molecules like plasma, red blood cells, protein. Okay, so these are not filtered, and what happens is that the efferent arterioles takes it out of the Bowman's capsule. Now the balance between the opposing and the favoring filtration, that is, the opposing is the ultrafiltration, the favoring filtration is the selective filtration. So the balance between the both of them is called the net filtration. So now if you want to calculate the net filtration with, say, afferent arterioles, which is 55, then we have the osmotic collider pressure, that is the pressure of the capillaries, and that is 30. Then we have the intracapsular pressure, and the intracapsular pressure is 50. So when you minus the afferent, the osmotic collider pressure, and the intracapsular pressure, it's going to give you the efferent pressure. So the efferent pressure equals to 10. So now we talk about the glomerular filtration rate. So the glomerular filtration rate is the total amount of tubular fluid that is being produced by the nephron of both kidneys per time. So the normal glomerular filtration rate is 180 liters in 24 hours. So we have 125 mils, that's milliliters, in a minute. Now the total amount of urine formed is about 1 to 1.5 liters a day. Now that is all for the glomerular filtration. So we talk about the tubular reabsorption. So the tubular reabsorption takes place in the tubules where essential nutrients are being reabsorbed back to the blood, so the body won't lack in its nutrients. So nutrients like glucose, amino acid, etc. are reabsorbed in the blood. So it takes place in the proximal convoluted tubules. It also takes place in the loops of Henle, the distal convoluted tubules, and the collecting duct. Now let's talk about the proximal convoluted tubules. Now the proximal convoluted tubules being the most coiled portion of the tubules. So 65% of the tubular reabsorption takes place in these proximal convoluted tubules. Now another thing we need to know about the proximal convoluted tubules is that it has a mitochondria that releases ATP for its activities. Now let's talk about the substances that are being reabsorbed in the proximal convoluted tubules. So we have sodium. Sodium is actively reabsorbed. Then we have H2O, which is water. So water is passively reabsorbed. Now we have chloride. Chloride is actively reabsorbed. So we have bicarbonate. Bicarbonate is passively reabsorbed. Then we also have glucose. Glucose is actively reabsorbed. So we have amino acid. Amino acid is actively reabsorbed. We have the urea. So urea is passively reabsorbed. And we have the creatinine. So the reabsorption of creatinine is entirely under the action or the influence of the calcitonin. Now, once all these substances are being reabsorbed, the tubular fluid in the proximal convoluted tubules becomes isotonic. So now we talk about what happens in the loops of Henle. So remember that the loops of Henle is divided into three. We have the descending loops of Henle, we have the apine band, and we have the ascending loops of Henle. So what happens in the descending loops of Henle? Now remember that sodium was actively reabsorbed. So the sodium that was actively reabsorbed now is secreted back into the tubules. Now the water that was passively reabsorbed in the proximal convoluted tubules is now actively reabsorbed. And now once this happens, the tubular fluid is going to become highly concentrated. So imagine salt is much in a particular place and water is out, so it's going to be salty. So I'm explaining it that way. So it's going to be highly concentrated, and it's going to lead the tubular fluid in that part to be hypertonic. Now we go over to the hairpin bend. So the hairpin bend, what happens in the hairpin bend is that as sodium is being secreted back, chloride also gets secreted back into the tubules. Now sodium and chloride being secreted back at the same time leads to what we call a countercurrent. So if you are asked, where does the countercurrent take place? It takes place in the loops of Henle, particularly the hairpin bend. So now we talk about what happens in the ascending loops of Henle. So we say that the ascending loops of Henle is impermeable to water. So it causes the reabsorption of water to stop, and the reabsorption of sodium begins. Remember that water begins to reabsorb in the descending. So now going to the ascending, it's impermeable to water. So the reabsorption of water will stop. So remember that the water was being actively reabsorbed in the descending, and sodium was secreted back. So that sodium that was secreted back now at this ascending will now be reabsorbed because the reabsorption of water has stopped. Now let's talk about what happens in the distal convoluted tubules. So now the distal convoluted tubules is now permeable to water. So remember the ascending loops of Henle was impermeable to water. Now the distal convoluted tubules is permeable to water. Now why is it permeable to water? Is because of the principal cells in the distal convoluted tubules. I remember the principal cells in the distal convoluted tubules is a receptor for ADH, which is the antidiuretic hormones. So the antidiuretic hormones act on the distal convoluted tubules and cause it to allow the flow of water. So now the next thing that happens there is that chloride becomes passively reabsorbed. The next thing that also happens there is that potassium is going to be secreted back into the tubules in place of the sodium. Now, what happens next is that hydrogen ions, this is when the kidney maintains acid-base balance. It primarily takes place in these distal convoluted tubules. So the hydrogen ions is going to be added back into the distal convoluted tubules depending on the acidity of the tubular fluid. So its work is just to come and make the tubular fluid to be stable and not acidic. So now, once all this happens, the tubular fluid in the distal convoluted tubules becomes isotonic. Now, take note that proximal convoluted tubular fluid was isotonic. Loops of Henle was hypertonic because of sodium, because of salt. Now, because of this adjustment and all that in the distal convoluted tubules is now back to its normal state, which is isotonic. Now, let's talk about what happens in the collecting dot. Now, the collecting dot is a tube that collects tubular fluid from different nephrons, and the tubular fluid in the collecting dot is called urine. It is impermeable to water. So it is impermeable to water except it's under the influence of still the antidiuretic hormone. So antidiuretic hormone main work is to maintain fluid balance in the body. So if it's under that influence, it's going to lose out water. So water is going to be reabsorbed in the collecting dot. Now, the composition of this urine form is 96% of water, 2% of urea. Then we have 2% of inorganic substances. So these inorganic substances include the urea, uric acid, the creatinine, the ammonium, the sulfate, the oxalates, etc. So that is for the tubular reabsorption. Now we talk about the tubular secretion. Now, the tubular secretion primarily takes place in the distal convoluted tubules and the collecting duct. So what happens is that substances are being secreted into the tubules to enable the elimination of waste. Now, there are two ways of tubular secretion. It's either the walls of the tubule secrete into its lumen, or the peritubular capillaries secrete into the lumen of the tubules. So we said a lot about this, what was secreted, like let's say the potassium that was secreted back. So all those we said that was secreted back in tubular reabsorption, that's what we can still say that's for tubular secretion. Now, the urine formed is clear and amber in color due to the presence of urobilin. So urobilin is this beribulin is when this red blood cells breaks down. So it breaks down and it's passed out through the urine as urobilin. Then it's passed out through feces as tecobilinogen. So that is all for the urine formation. So I guess this, I hope this video was a bit helpful. Do well to form your notes and do well to like, share, and follow for more health education. Thank you. Bye.